Okay, Chapter 12 and 13. Chapter 12, there's not a whole lot I want you to know. Mostly just get some familiarity with some of these key terms, and we'll go over some of these. Acquired resistance, intrinsic resistance, how are they different from each other? What is an antibiotic? What are beta-lactam antibiotics? What is a beta-lactamase? And what does synergy mean? So antibiotics and antimicrobial agents are chemicals that we use to either kill or suppress the growth of microorganisms. They can be natural, they can be semi-synthetic or synthetic molecules, and we're not going to go into depth about what exactly that means, but keep in mind there's different categories of antibiotics, there's different generations of antibiotics, so they've evolved over time to meet the increased resistance that we're seeing in these organisms. So resistance can be either intrinsic or acquired. So intrinsic resistance is naturally found in the bacteria. So usually all of your genus is going to be sensitive or resistant to that particular antibiotic. For example, Klebsiella is resistant to ampicillin. We don't need to test it. It's always resistant. It's intrinsic resistance. It's built into its DNA. Acquired resistance is an acquired resistance to an antibiotic that is from an outside source. So, for example, passing some DNA from one organism to another organism. The new organism acquires that resistance from the other organisms. So it's passed from one to another. So antibiotic targets and mechanisms of actions, we're not going to go into how all these different antibiotics work. We will talk about beta-lactam antibiotics and an enzyme called beta-lactamase that is a resistance enzyme for beta-lactam antibiotics. So basically that works on the cell wall of the organism. So we talked about the peptidoglycan in the cell wall of bacteria. These organisms interfere with the synthesis of this cell wall. So by disrupting the formation of the cell wall, the organism is it's at a disadvantage. It can't function without its cell wall. It just ruptures. And they can also work by interrupting some metabolic functions. So how the organism synthesizes proteins, or how does it metabolize nucleic acids, or somehow interfering with the inner workings of the cell so that it can't function. So basically, what I want you to know from this big table is cell wall synthesis inhibitors. So your beta-lactam antibiotics, and that's your penicillins. Probably everyone has heard of penicillin or ampicillin, oxacillin. These are beta-lactams, and they interfere with the synthesis of the cell wall in bacteria, and so inhibit them from growing. Hopefully this looks a little familiar. This is the NAG and NAM. So this is the peptidoglycan layer, and it's just these repeating units of NAG and NAM. And the beta-lactam antibiotics, they sort of put themselves in this repeating subunit and interfere. It's not able to build its lattice work and build its cell wall the way it needs to because this antibiotic is interfering. So these are your basic antibiotics, and I mostly just want you to think of penicillin and ampicillin when I say beta-lactam. And all of them have this basic subunit of a beta-lactam ring. You can see it right here. So that's why they're all called beta-lactams. They have this similar structure somewhere inside them. So beta-lactamase, we'll talk about a beta-lactamase enzyme that breaks this ring apart. So if an organism produces the beta-lactamase enzyme, it's able to break up this beta-lactam ring, and that essentially makes the penicillin inefficient. So it's resistant to any kind of beta-lactam because it produces the beta-lactamase enzyme. And we'll talk about how we test for production of that enzyme in a bit. Synergy is referred to, it's about combining the effects of multiple antibiotics for a greater impact or an enhanced activity. So you might think that using 2 antibiotics would work better for a patient. They would work synergistically and have an added effect and work better, but that's not always the case. There are some antibiotics, if they are used together, that can have a detrimental effect on the patient, or they won't work as well. And we'll talk about some inducible resistance to clindamycin in a little while, and we'll see that these 2 antibiotics may not work together effectively, so you don't want to use them together. Intrinsic resistance and acquired resistance, again, we talked about that just a minute ago, whether it's in the bacterial DNA or did it acquire it from an outside source. There are a lot of different mechanisms for acquiring this resistance. Maybe the bacterial target for the drug, maybe it doesn't bind very well, so it lacks affinity for the drug. If the drug can't bind to the organism, then it can't get inside and do what it needs to do to kill the organism. It can prevent access to the drug to the site of action. So maybe there's something the organism has developed that keeps the drug from getting into the cell wall or from passing through a membrane. Biofilms are like this thick film of growth. It's that can keep drugs from getting into the bacteria that are in these films. Efflux can have an impact on the resistance, whether the drug is secreted from the organism. Enzymatic inactivation, does the organism have a particular enzyme that destroys the drug or inactivates it? That's an example of beta-lactamase. So again, here's penicillin with its beta-lactam ring. And if the organism produces the beta-lactamase enzyme, it breaks that ring and makes the antibiotic inactive, inefficient. It doesn't work. This is just kind of a picture of an acquired resistance. So this is passing some kind of genetic material from one organism to another. And I saw it firsthand in a patient that had multiple infections. So she had a variety of different organisms that was causing bed sores and urinary tract infections. She had organisms in her blood. She was a really sick lady. And so she had multiple organisms on her body that were causing an infection. She had vancomycin resistant Enterococcus that was in a bed sore, and then she also had methicillin resistant Staph aureus in her bloodstream. So she has these 2 different organisms. Methicillin resistant Staph aureus had the gene for the methicillin resistance, and then this Enterococcus had another gene for vancomycin resistance. and what happened was they were in close quarters, they communicated with each other, and that Enterococcus passed that gene for vancomycin resistance to the Staphylococcus aureus, and what happened was the staph then acquired resistance to the vancomycin. So it was resistant to methicillin and vancomycin, and that is a really bad combination. There's not much that we can do for a person that has a Staph aureus that is methicillin resistant and vancomycin resistant. Those are heavy-duty drugs, and there's not much else left to treat a patient once they acquire both those resistance genes. So, Chapter 13, we're going to talk about different methods that we use in the lab in order to test bacteria for antimicrobial sensitivities. Okay, so take a look at these objectives. As you're studying for your test. So, antimicrobial susceptibility testing. This is done on bacteria that's isolated from our clinical samples, and it helps us to determine what agents will be effective in treating these infections. So, not all organisms that we isolate in the laboratory are going to require antimicrobial testing. So it's important that we distinguish pathogens from commensals, so we're not performing all of this sensitivity testing on organisms that are just normal flora. So there's different methods of performing this sensitivity testing or susceptibility testing. You can use a disc diffusion method where you place a disc on a plate of agar that's been inoculated with the organism, and the disc of the antibiotic on the disc is going to diffuse out into the agar and inhibit the organism. Or if the organism is resistant, the organism's going to grow up to the disc. There's also dilution methods where you can dilute, you can make dilutions of the organism, or you can make dilutions of the antibiotic itself and test the organism against those different dilutions of antibiotic. So we'll talk about a couple of these. There's a lot of different types of sensitivity testing out there, but we're going to focus on the major two concepts of disc diffusion and then your MICs. We're going to make a dilution of the antibiotic and see if the organism is sensitive or resistant to increasing dilutions of that antibiotic. So we'll use a standard guide that's published by the Clinical and Laboratory Standards Institute, so they determine what value of inhibition correlates to a sensitive or a resistant interpretation. And that kind of sounds like a jumbled mess, but let's get into how these methods are performed, and you'll see how we get to those interpretations. So why do we perform susceptibility testing? Because an isolate that is determined to be a possible cause of infection, we need to find out what that organism is susceptible to and what it's resistant to. So we don't always perform sensitivity testing on all bacteria, even all pathogens. So your group A strep that causes strep throat is universally susceptible to penicillin. So we usually don't perform sensitivity testing on group A strep unless the patient is allergic to the drug. Then we would have to perform. We'd have to find something else that we could use to battle that group A strep. Some factors to consider when you're performing susceptibility testing. What was the site of collection? Where was the organism isolated from? You might treat organisms isolated from a site that has a lot of normal flora differently than you would an organism that was isolated from a sterile site. The specimen quality, are there signs of poor specimen quality? You wouldn't have to set up sensitivities on a sample that is of poor quality. What's the host status? If the patient is immunocompromised, you may have to do some additional sensitivity testing. Or are they allergic to the drug? Or can they not handle a certain type of drug? So you might have to do some additional testing there. This is presence of other bacteria and the quality of the sample. Pure cultures generally indicate a good quality sample, so you would perform sensitivity testing if it's a good one. The presence of two different organisms suggests contamination, so we see this a lot in sputum samples or urinalysis samples, and in that case you may not have to perform sensitivity testing. And your policy for when you should and when you shouldn't perform sensitivity testing, it's all outlined in the facility that you would work at. So it can change depending on where you work. There's some the host status can influence the susceptibility testing. The presence of normal flora. In a normal immunocompetent patient would be considered a contaminant or normal, but normal flora in an immunocompromised patient may indicate an infection. Immunocompromised patients can't fight disease like someone with a good immune system, so they can get these infections with organisms that normally don't cause disease. So in that case, you should do some susceptibility testing. You may need bactericidal concentration. So there's two key terms here. We have bactericidal, which is the level of antibacterial that kills the microorganism. And then we have bacteriostatic, which is an antibiotic that just inhibits the growth of bacteria. It doesn't kill the organism, but it stops it from replicating. Allergies may require you to perform sensitivity testing. We said if a person is allergic to penicillin, they would need a different drug for their treatment if they have group A strep causing them strep throat. So in that case, you would have to perform some sensitivity testing. Once you've decided that you need to perform sensitivity testing, there are different batteries that you would perform based on the group of organisms that you're working with. So usually you have a gram-positive battery and a gram-negative battery. So you're going to use certain antibiotics against gram-negative organisms, and you're going to use a separate set of antibiotics for gram-positive. And that's because antibiotics work differently. Some antibiotics would work against a gram-positive organisms, but they won't work against a gram-negative organism. So each facility has developed these different battery of antimicrobial agents that gram-positive organisms are tested against, and then a completely different set of gram-negative antibiotics. And there may be some overlap between the two, but you'll see some differences. They may have a completely separate battery for an organism isolated from the urinary tract. So keep that in mind. Some drugs are only used for urinary tract infections, so they would be incorporated in this sensitivity panel, but not incorporated into a panel used for, say, a wound or a sputum. There's a lot in your textbook about how to report sensitivity results, and we're not gonna go into depth about that. But your primary objectives at any clinical site is to report the least toxic antibiotics, the most cost-effective antibiotics for the patient. You would rather test against an antimicrobial that might be $50 a bottle as opposed to something that's $300 a bottle. What's clinically appropriate for whatever agent you would treat an infection with Serratia marcescens differently than you would an infection with a Staphylococcus aureus. So use what is appropriate for what's causing the infection. And then you want to avoid using very expensive broad-spectrum antibiotics. Broad-spectrum antibiotics, if you think about the name, it's going to work against a wider array of organisms as opposed to a narrow-spectrum antibiotic that's only going to work against a smaller number of organisms. So your broad-spectrum antibiotics, they're very costly and expensive. They can also promote resistance among organisms. They can kill off a lot of your normal flora, causing you to get an opportunistic infection. So ideally, we want to get patients on a narrow-spectrum antibiotic. Okay, once you've determined which antibiotics that you need to test, you're ready to perform your sensitivity testing on your test organism. So there's different methods. We're going to talk about a disc diffusion method first. So to begin with a disc diffusion method of susceptibility testing, you begin by preparing an inoculum preparation. And this is basically a standard inoculum or suspension of your test organism that is matched against a known standard, which is your 0.5 McFarland standard. That's a turbidity standard. So basically what you do is pick 4 or 5 isolated colonies from a non-inhibitory agar, so a non-selective agar like a TSA plate or a sheep blood agar or a chocolate agar plate. And you want to emulsify those colonies into some broth or some sterile saline. And it's going to be, the solution is going to become hazy. And you want to match that turbidity or the haziness to a known standard, the 0.5% or 0.5 McFarland standard. Okay, you do have the amount of inoculum or the amount of bacteria that we use to prepare this this test has to be standardized, and that's because if we use too little bacteria or too few bacteria, we can see false susceptibilities, or the zone of inhibition that we'll see will be too large and will report drugs as being susceptible when they may in fact not be susceptible. If our inoculum is too heavy and we have too much bacteria, we can see a false resistance. So our zone sizes are going to be too small, and we would report more resistance. And we'll see how that is, how we determine that in just a minute. So once you prepare your standard, you're going to vortex your 0.5% and prepare a spread plate. And you can compare your McFarland standard by holding up a piece of notebook paper that has lines on it, and compare that with your standard to see if your turbidity is matching. You can also use a spectrophotometer, but we're not going to do that in our lab. So here's an example of the McFarland standard. This is the 0.5 McFarland standard, and this is a 2 McFarland standard. So our inoculum, when we suspend it in some saline or some broth, we want it to match this standard here. And notice this one is a lot heavier than the 0.5. So we want a very light, hazy suspension of our inoculum. And we're going to do a spread plate technique of our inoculum. There are a couple of videos that I've posted under this week that illustrates how to do a spread plate for antimicrobial testing. And basically what your goal is to have a lawn of growth or confluent growth over the entire surface of the plate. You don't want any gaps in growth. You want a nice long lawn of growth. Some dilution susceptibility testing methods are available, and these methods allow you to determine the minimum inhibitory concentration of antibiotic. So that is the lowest concentration of antimicrobial agent or antibiotic that is required to inhibit the growth of bacteria. Okay, very important definition there. We also have the minimum bactericidal concentration, and we're not really going to go into depth or do any kind of testing on the MBC, but this is the lowest concentration of antibiotic that kills 99.9% of the test bacteria. So if you take a bit of that suspension and put it on a plate, there should be no growth whatsoever. So a standard inoculum of the bacteria is placed in diluted concentrations of the antibiotic and incubated. So interpreting the dilution method, this stands in place for MIC testing and for disc diffusion testing. So basically the value of the MIC or the value of the zone of inhibition on a disc diffusion plate is compared to a known value for resistance. And there's a manual in the laboratory that we use to determine those known values. So you look up your organism, you look up your antibiotic, you find that MIC or the zone of inhibition, and that's going to correlate to either susceptible or intermediate or resistant to that particular antibiotic. And I know that might be a little bit fuzzy now, but we're gonna apply it in lab. So here's some examples of minimum inhibitory concentration standards for different organism groups. So here we've got a bunch of different organisms and their MICs for ampicillin. So if you're testing Enterobacteriaceae, if your MIC is less than or equal to 8, that indicates the organism is susceptible to that antibiotic. If your MIC is greater than 32 or greater, that means that the organism is resistant to that antibiotic. All right, and if it's anywhere in between, then you would be an intermediate. So you can't really say it's susceptible or resistant at that point. So you can see we've got different, depending on what organism you're performing your testing on. Is it a staph? Is it an Enterococcus? Is it a Haemophilus? You would use those MIC reference standards based on what you're testing. Okay, there's also some Broth-Microdilution Tests. We're not going to be diluting broths, but we will see a multi-well dilution of antibiotics on a plate of a bunch of micro wells. So it's handy to test a variety of antibiotics against the same organism at one time. So you're using less media, less antibiotics. It can be automated. You're using less organism. There are some sources of errors depending on the method that you're using. So trailing or skipped wells can be seen. There's different causes for these It might be contamination. It might be poor inoculation of the wells. It might be poor dilution of the antibiotics in the wells, or a resistant subpopulation. It could just be something that just sort of happens. So we're not going to go into depth about those irregularities. You can see that when you go to your sites. It's not something that occurs a lot, and the procedure manuals are pretty clear on how to handle them. Okay, so disc diffusion testing. We also call this Kirby-Bauer test. And what you do is you take a swab of your 0.5 McFarland standard inoculum, and you swab it over the entire surface of the plate. And we usually use a Mueller-Henton plate for this. And then once you've swabbed your entire plate with your organism, then you add a paper disc that contains the antibiotic on it, and you incubate it overnight. All right. When you incubate it, the antibiotic is going to diffuse from that paper disc out into the agar. And so you'll have a very strong concentration very close to the disc, and decreasing concentration of the antibiotic the farther away from the disc that you get. And so what you get is a circle or a zone of inhibition around the disc. And so you measure the diameter of that zone, and you're going to compare it to the standard manual that we have in lab to determine if that zone size correlates to a sensitive or resistant interpretation. You read these zones of inhibition as in millimeters. So here's an example. This is a Mueller-Henton disc, and we swabbed the entire plate with our 0.5 standard inoculum, and we got a lawn of growth. So once we swabbed it, we placed each of those antibiotic discs on the plate, and you can see some of them were very successful at inhibiting the growth. We have a very large zone of inhibition, and some of these on the top right, we have growth all the way up to the edge of the disc. So that indicates that the organism was resistant to that particular antibiotic. So like I said, we put the antibiotic disc on the plate, and the antibiotic diffuses out onto the plate, so you have very strong concentrations the closer you get to the disc, and it gradually decreases in concentration the farther away from the disc you move. This is a stamper that you can use. You place the stamper over the Mueller-Henton disc and push down on that top lever there, and it places all of those discs on the agar equal distance from each other, so that you're not too close. You don't want to get antibiotics too close to each other, or you'll interfere with the zone of inhibition. So disc storage, you want to make sure that you either keep them in the refrigerator. They're only good for 1 week. Otherwise, you want to keep them in the freezer. They have to be FDA approved discs. The agar depth is standardized between 3 and 5 millimeters thick, and the pH is between 7.2 and 7.4. So if the discs aren't stored properly, if they're past their expiration date and they're not working well, you can have falsely decreased zones of inhibition. If your agar depth is too thick, then you're also going to have falsely increased zones of inhibition. If your agar is too thin, you're going to have falsely decreased zones of inhibition. If your inoculum size is too light, your zone of inhibition is going to be too large. If your inoculum is much too heavy, then your zone of inhibition is much too small. So if you kind of think about that zone of inhibition and how the antibiotic diffuses through the agar. If we change something, if it's either the inoculum's too weak or too strong, or the antibiotic is not strong enough, that zone of inhibition is going to change. It's either going to increase or it's going to decrease. I do know that there are a couple test questions about if your agar depth is too thick, what would that do to your zone of inhibition? Or if your inoculum is too thin, what would that do to your zone of inhibition? Would you have false resistance or false susceptibility? Let's see, the media formulation. Our basic media for antibiotic testing is the Mueller-Hinton disc. Now, for some of your fastidious organisms, it may require a modified Mueller-Hinton, something that has some enriched properties to it to grow those fastidious organisms. So keep that in mind. Your lawn of growth should be completely surrounding or covering that Mueller-Hinton disc. So if you have large gaps in your lawn of growth, it's going to interfere. Your zones of inhibition are going to be disrupted. Your zone of inhibition shouldn't contain colonies inside that zone. So if you've got a couple isolated colonies inside that zone of inhibition, they could be strains that are resistant to that particular antibiotic, or it could be contaminated with another organism. So you generally want to see a nice clear zone of inhibition. Proteus can be a little bit difficult. We're not going to see a Proteus in the lab, but it does swarm over the entire plate. So interpreting the zone size can be a little bit trickier. Here's an example of a poor lawn of growth for your organism. We got lots of gaps here. We've got isolated colonies. We don't want isolated colonies in here. We want confluent growth. We want a nice mat or lawn of growth for our organisms. So they might have used an inoculum size that was too small, or they didn't streak it well enough when they were preparing it. Here's ideal. We've got a nice lawn of growth between our discs, and this person is measuring in millimeters the diameter of the zone of inhibition. So for each antibiotic, you're going to measure that diameter, and you'll look up that diameter in our reference manual to determine if it's sensitive or resistant. Here, this one has growth all the way up to the disc, so I don't have to measure anything. I know that that drug is resistant. This one also resistant. These zone sizes are very, very small, and so they'll probably be resistant as well. So say we measure our zone of inhibition. Say we're measuring this one here. Say we get 26, okay? Say that drug was ampicillin, and we're testing E. coli, which is an Enterobacteriaceae. So we're testing E. coli against ampicillin. The disc was a 10 microgram strength of ampicillin, and our zone size was 26. What interpretation would that correlate to? Well, here's our zone diameter. So this is for disc diffusion. So I would use these parameters here, and 26 is greater than 17. So that would correlate to a susceptible interpretation. If I was doing MIC testing, I would use this side of the table. But disc diffusion testing, you measure the zone diameter, the zone of inhibition in millimeters. So say my E. coli, when I tested it against gentamicin, say it was 8 millimeter diameter. Well, that would correlate to an interpretation of resistant for gentamicin. Like I said, for some organisms, we modify the Mueller-Hinton a little bit. For Streptococcus pneumoniae, we use a blood agar plate. So it's a Mueller-Hinton with some sheep blood mixed into it. Let's see, Haemophilus is another fastidious organism, so we use Haemophilus test medium, which is just a Mueller-Hinton agar base, but it's also enriched with some factors that Haemophilus needs in order to grow. Some of your Haemophilus species do produce beta-lactamase. Remember, beta-lactamase is an enzyme that breaks down the beta-lactam ring in beta-lactam antibiotics like penicillin and ampicillin. Okay, so we have some testing methods to test for that beta-lactamase enzyme itself. So 20 to 50% of your Haemophilus species do produce this beta-lactamase. So in that case, if it does produce beta-lactamase, we can't use penicillin or ampicillin to treat a Haemophilus infection. Some other sensitivities to keep in mind, vancomycin and methicillin resistance in Staphylococcus aureus. These are really important. We use oxacillin or cefoxitin disc, to test for methicillin resistance in Staph aureus. We can either do a disc diffusion method where we plate the organism on our Mueller-Hinton disc and then place an oxacillin disc on that plate and measure the zone of inhibition. There's also plates that are infused with methicillin or oxacillin, and if we inoculate those plates with the organism, if it grows, it indicates it's resistant to the antibiotic in that plate. Some inducible clindamycin resistance can be measured in Staphylococcus. So this involves erythromycin and clindamycin. They're placed 15 millimeters apart on our Mueller-Hinton disc. Erythromycin-resistant isolates that may appear susceptible at first, we need to perform this inducible resistance. So it's called the D test when we place these two antibiotics 15 millimeters apart. Usually they're much farther apart. And basically what this means is if these two drugs interact together, there may be an inducible resistance. They may not work well together. So we're testing for that with the D test. And if we place these two antibiotics close together, if we see this flattening zone between the clindamycin and erythromycin, it forms a D shape instead of a nice round zone of inhibition. It's inducible resistance. So in that case, we would report both erythromycin and clindamycin as resistant. And usually, most labs automatically test for this. So here's an example. We put our erythromycin disc 15 millimeters close to our clindamycin disc and incubated it overnight. And instead of having a nice round zone of inhibition, we have this flattened edge between the two antibiotics. So it's in the shape of a D. So the D test is positive. We're positive for the inducible resistance to clindamycin. So we cannot use these two drugs together. We report them both as resistant. It's very important that we detect some vancomycin resistance, particularly in Enterococcus species. So it can also be done on a disc diffusion method, or we can buy these specially prepared plates that are infused with the antibiotic. We can use these same plates to screen Staph aureus for vancomycin resistance. So this is a really big hot topic, testing patients to see if they have a vancomycin resistant organism. That way we know that we're going to have to use something stronger than vancomycin, and there's really nothing much out there stronger than vancomycin. So it's important that we catch it if it's there. There are some automated methods available for susceptibility testing, and a lot of places are using these automated methods instead of a manual disc diffusion method. So basically you inoculate a plate that has a lot of microwells in it, or somehow you inoculate some type of compact, and the analyzer reads the susceptibilities for you. The E-test is an interesting kind of twist on disc diffusion. Instead of inoculating a plate with a antibiotic disc, you use a strip that is inoculated, a strip that has the antibiotic on it. And instead of having a certain concentration of antibiotic like the discs, you have an increasing concentration of the antibiotic. So it has a varying dilutions of the antibiotic. And what you want to do is read where the growth touches the strip. So I believe I have a picture. Here we go. So we inoculate our plate just like we would for a disc diffusion method. We want a lawn of growth, and we place our E-strip on that growth and incubate it for 24 hours. And so we have a weaker concentration of antibiotic down at the bottom of the strip, and it increases in concentration towards the top, towards the GM. So what results is an elliptical shaped pattern, and we want to read where this elliptical touches the strip. So here on this side it touches between 0.5 and 0.75, and then on this side it's touching about the same, between 0.5 and 0.75. So that would be interpreted, I would take the higher of the two, so I would report 0.75 as my minimum inhibitory concentration. So MIC. Beta-lactamase tests, which test for the beta-lactamase enzyme that breaks down the beta-lactam ring. A nitrocephin disc is a beta-lactamase test, and we'll do some of these in lab. Basically all you do is smear some of the colony onto the disc, and if a red color or a pink color develops, that organism produces beta-lactamase. So you would not report, so penicillin would be ineffective, ampicillin would be ineffective. Okay, and we commonly do this on organisms like Neisseria or Haemophilus. One advantage for this method, it's very rapid. You don't require overnight incubation. It's a lot easier and a lot quicker than the overnight disc diffusion method. So here it is. We've got our beta-lactamase disc, and we smeared our organism colony onto this disc, and it turned pink. So we can report that penicillin and ampicillin, any kind of beta-lactam antibiotic, is going to be inefficient. On the right side, we have a negative result. This is just a little bit about, there's a lot of different special methods that are available for antimicrobial testing. Basically, I wanted to point out that there are some molecular methods available, and so we can determine if an organism is methicillin-resistant Staphylococcus aureus within 4 hours, as opposed to waiting for an overnight susceptibility test, which could take 48 hours after you see a patient. So instead, it's a molecular test that's looking for this MEK-A gene in Staphylococcus aureus, and it can flag a result in as little as 4 hours, and so you can treat your patient much quicker. You can know that they have this resistant organism much faster, and hopefully have better outcomes with that molecular method. So that completes the presentation or the lecture on this topic. I do suggest that you go to Blackboard and check out the videos. I have a video of preparing a 0.5% McFarland standard. Check that out. You can use either saline or broth to prepare this standard. We have a Kirby-Bauer disc diffusion video. So this is a guy preparing a Kirby-Bauer test and putting the discs on the plate and incubating them. He doesn't prepare a 0.5% standard, but we do in the laboratory. Our testing is much more structured than what he's performing there, but it's the same idea. I also got a video on the E-test, how to perform that, very similar to a disc diffusion. It's just you're putting a strip on the plate instead of a disc. Here's a picture of a strep pneumo that we use to test with the E-test. And ideally, you don't want all these bubbles. You want to get those out. It should look like this when you put your strip down. So if you put it on there and you see some bubbles, you want to use some tweezers to try to get those bubbles out. But you want to read where the growth starts, not where the hemolysis starts. So you want to kind of, it looks like it's kind of growing between 0.64 and 0.94. So I would report 0.094 as my MIC. And then I would have to look that up in my standard manual to see if it was susceptible or resistant. Here's an example of a D test. Is it negative? Is it positive? What are we looking for in the D test? AZM is basically your erythromycin, and DA is your clindamycin. So we're looking for a flattened zone between the clindamycin and the erythromycin. I don't see a flattened zone. This kind of comes to a nice rounded point here. So I would call that negative. I would measure the diameter between these two zone of inhibitions and record those diameters, and then look those values up in my reference manual to see if it's resistant or susceptible. Here's a good example of a positive D test. So my zithromycin, I don't really even have to look it up. It's got a very, very small zone. It's probably going to be resistant. Here, my clindamycin, notice I have all this zone of inhibition, but all of a sudden it flattens out right here. It's in the shape of a D. So I don't have to measure that zone. I can call it resistant. If that flattened edge is there, it's resistant. Here's an example of a microscan panel, and you'll get to interpret some of these in lab. It has biochemicals in the top half, and then it has these antibiotics on the bottom half. So if you note, if you look really hard, we've got, here's AZT. So whatever antibiotic that is, we've got, what is that, a 4 and 16, or an 8 and 16. So this antibiotic, this well, is correlates to maybe 8 micrograms of antibiotic, and this is 16 micrograms of antibiotic. And notice it's a little hazy there, so the organism looks like it's resistant to both of those strengths of antibiotics. So my MIC is somewhere greater than 16. Here this is CAZ, and we've got increasing concentrations, 1, 4, 8, 16, and I've got a little bit of growth in my first well. So what I'm looking for is my MIC, the minimum concentration of antibiotic that inhibits growth. And so the smallest amount of antibiotic that inhibits growth here is 4. So I would look a 4 MIC up in my book and determine if that correlates to a sensitive or resistant MIC. Here this is CFT. I've got 2, 4, maybe 2, 8, 16, 32. It looks like these first 3 wells have growth, and this last well of 32 has no growth. So my MIC for this antibiotic would be 32. It's the smallest concentration of antibiotic that inhibits growth. So that's basically what you'll be doing in lab, recording the MICs and looking them up in the book. So keep in mind if you have growth in all wells, so all the way up to the highest concentration of antibiotic, your MIC is greater than whatever that value is. So in this case it was greater than 16. If I have no growth in any of my wells, for example this is LVX, levofloxacin, I've got nice clear wells in both. So my MIC could be 2, it could be less than 2. So I'm going to report less than or equal to 2 for levofloxacin. Up on the top left are your control wells. You have a C well and a G well. You should always have growth in your growth well, and you should always have no growth in your control well. So take a look at those 2 wells before you get started. Make sure your control wells are good, and then start recording your MICs.